All right, class, in this recording, we are going to focus on the innate or the non-specific immune system. Our non-specific immune system is going to be our first and second line of defense. Our first line of defense is very, very non-specific. It's just going to be physical barriers and chemical barriers that keep the bad guys, that keep the pathogens from getting into our bodies. Our second line of defense is going to be a cellular line of defense. We are going to have cells within our bodies that will remove generic pathogens. These cells are not pathogen specific. They remove all bacteria or all viruses, and generally speaking are equally effective on all pathogen types. So our first line of first line of defense, which is part of our non-specific immune system, is going to be the physical barrier to entry. That second line of defense is going to rely on some broad categories of white blood cells, the inflammatory response, and some categories of proteins, particularly complement and interferon proteins. So let's talk about innate immune defenses. These are all going to be broad defenses that don't act on any one specific species of pathogen. They act on multiple species of pathogens. We have the physical barriers to entry, aka our skin and mucous membranes. We are going to have protective films and protective proteins within our body. So we have the complement proteins and the interferon system within our blood plasma. We are going to have neutrophils and monocytes and macrophages within, and I should also mention dendritic cells, within our tissues. And those are going to phagocytize pathogens. We also have natural killer cells that are going to destroy intracellular pathogens. And then we're also going to have the inflammatory response. Our inflammatory response is going to increase the vasodilation or the permeability of our blood capillaries and make it easier for white blood cells to get to the action, to get to the battle scene. We also will have a system of chemical signals known as the cytokine system. These cytokines are the emergency alarm. They sound the alarm for our immune system to attract more neutrophils and monocytes to the site of infection. So let's talk about the physical barriers in more detail. Let's look at the skin. The skin is made of a stratified squamous, cornified epithelium. That's a nice way of saying that we have many layers of flattened cells that are filled with abrasion-resistant proteins. And these cells are designed to slough off. You can think of them as a blade of armor for those of you who are really into Star Trek Voyager or the Star Trek universe, and I'm letting my inner nerd show right now. We also are going to have some physical barriers in terms of the mucous membranes, tears, and saliva within our bodies. We are constantly flushing liquids out of our body, and as we flush those liquids out of our body, we are going to flush microbes with those liquids. Within our mucous membranes, we contain a lot of immunoglobulins that help to neutralize pathogens, and we'll have lots of mucous membranes anywhere something from the external environment gets to the inside of our body. So let's focus on the digestive tract and the respiratory and urinary tracts. We also are going to have resident bacteria within our bodies. These resident bacteria within our bodies are going to outcompete the pathogenic. You can think of these as the good guys, so to speak. They're good for us in the fact that they take up space and make it difficult for the pathogenic bacterial species to move in and make us sick. Hold up, Butler. I'm eating candy. I love Christmas candy. Anyways, let's talk about the chemical barriers in our body. Our chemical barriers in our body include the secretions of our oil glands or sebaceous glands. The sebum that comes from these sebaceous or oil glands is going to contain antimicrobial proteins and antimicrobial chemical compounds. One particular antimicrobial protein is called lysozyme. Lysozyme is an enzyme that causes bacterial cells to lyse or pop like a balloon that's overinflated with water. We have lots of lysozymes present in both our saliva, tears, and sweat. It's worth emphasizing or mentioning that we as human beings have relatively high concentrations of lysozymes within our saliva. So the next time you get a paper cut and you have that instinctual urge to suck on your fingertip after getting the paper cut, that's not necessarily a bad thing to do because you have antimicrobial proteins present in your spit. So the next time your friend skins their knee, do them a favor and spit on the wound. It may be gross, but you're putting lysozymes on their wound to help reduce the bacterial count. It's also worth emphasizing that dogs have very low concentrations of lysozymes in their saliva, and consequently the bacterial concentration within dog saliva is much higher, and that's one of the reasons why dog breath is particularly bad smelling. If we look at the stomach and vagina, both the stomach and the vagina have an acidic environment which inhibits or hinders the growth of many microorganisms, particularly in the stomach where we have a pH of 2 within our stomach chyme. That's going to make it impossible for most bacterial species to grow. We also are going to have some phagocytotically active white blood cells. Whew, phagocytotically active. That's a $10 word if I ever saw one. When I look at this term, phagocytotically active, the root word phago means to eat, cyto means cell, and active means, well, it's active, it's doing it. So a phagocytotically active cell is going to be a cell that is actively eating or pursuing the consumption of other cells. So if you've seen that 1950s classic movie, The Blob, where there's this giant blob swallowing people within a rural country town, that's kind of like what the white blood cells, or at least the phagocytotically active white blood cells in our bodies do. These phagocytotically active white blood cells are going to be primarily the macrophages, which are derived from the monocyte line of cell, white leukocytes or white blood cells. So monocytes turn into macrophages. To a lesser degree, neutrophils. I primarily consider the neutrophils the kamikaze pilot. Yes, they are slightly phagocytotically active, but neutrophils primarily function to kill the cell without having phagocytosis occur by the release of proteases, or enzymes that digest the cell wall and the cell membrane of the bacteria. Both neutrophils and macrophages will leave the bloodstream, leave circulation, and move into the tissues of our body to pursue the site of infection. How do they know when they need to leave the bloodstream? I'm glad you asked. These neutrophils and macrophages know when to leave our bloodstream because they follow chemical messengers released by the dying tissues and cells of our bodies. These chemical messengers are called cytokines. You can think of them as smoke leading to a fire. And if a firefighter is in a neighborhood and they see a bunch of smoke rising up in the air, they know, hey, I should probably go to where the smoke is coming from because there's probably a fire there I should put out. In the same way, when a neutrophil or a macrophage is exposed to cytokines, they are attracted to those cytokines because cytokines imply some kind of infection taking place. These neutrophils and macrophages are also an important part of the inflammatory process. If you look at the inflammatory process, by causing our blood capillaries to dilate and become more permeable, we have more fluids leak out. That causes inflammation, but it also makes it easier for these white blood cells to get to the site of infection. Let's talk about the inflammatory response in a little bit more detail. There are some hallmarks associated with inflammation. All of these hallmarks can be linked back to vasodilation. As we have vasodilation, or the blood vessels get wider and more permeable, we are going to have more liquid leak out of the blood vessels. That causes swelling. As we have increased blood flow, that causes more red blood cells to be there. That causes redness. As we have more blood delivered to the extremity from the core of the body, the blood will carry heat with it and will cause an increase in warmth at that area where there's inflammation. And then finally, as we have the swelling and the vasodilation occurring, the same chemical receptors that cause the vasodilation will also activate pain receptors within those tissues. The primary chemical receptor that causes, or chemical messenger that causes vasodilation and inflammation is histamine. Histamine is a neurotransmitter that's released from mast cells. Mast cells are derived from basophils. And this histamine is going to cause the blood capillaries to become permeable and dilate. If we look at this increased blood flow, we're also going to have more white blood cells delivered to the site of infection thanks to inflammation. And inflammation is a double-edged sword. It's both good and bad. It's good because it delivers more white blood cells to the site of infection, but it can be bad because too much inflammation will actually pinch off the blood vessels and impede tissue healing because we don't have enough nutrients being delivered to those tissues. So a little inflammation is good, a lot of inflammation is bad. Our inflammatory response can be short-lived, and short-lived inflammatory responses, generally speaking, are fairly good for us. Long-term inflammatory responses, though, are not good for us. At that point in time, blood flow is impeded from excess tissue swelling, and we don't have proper nutrient delivery. The cytokines can call, as I mentioned before, will call in the white blood cells, particularly the macrophages, to the site of infection. So here's a generic puncture wound to our skin. At that puncture wound, we're going to have bacteria getting into the site of infection. We'll have a blood clot form because, hey, we need to stop bleeding. And that injured tissue and cell is going to release some histamine. Histamine is going to cause inflammation. And then we're also going to have some cytokines released. These cytokines that are released are going to attract more white blood cells that will exit the bloodstream and go to the site of infection to phagocytize the bacterium. Another line of defense within our nonspecific immune system is the complement and interferon protein system. These are both line 2, or part of line 2 of our line defense. We had the physical and chemical barriers for line 1 of our nonspecific immune system. Line 2 is going to involve some cellular and protein-based lines of defenses. Here's the protein-based. The complement system is fun. It's cool. It's a way to turn a bacterial cell into a wiffle ball, so to speak. We have a bunch of proteins floating around within our blood plasma. These proteins are collectively called the complement proteins. And once our complement proteins start to bind to a pathogen, more and more of them will form. This is a positive feedback loop. Once one complement protein binds, they all start to bind very rapidly. And as there's rapid binding of complement proteins to the bacterium, these complement proteins will perforate the bacterium. They will punch many hundreds of tiny little holes in the bacterial cell membrane. And by perforating the bacterial cell membrane, this causes the concentration gradients to no longer be maintained, and the bacterium swells up with liquid and pops like a balloon. We also have interferons. Interferons are a separate category of proteins, and these interferons are going to be made by a cell infected by a virus. What do interferons do? They send out an alarm to nearby cells within the tissue to say, hey, I'm infected by a virus. You should back away, and by the way, here's what the virus looks like. Now you can be temporarily immune to the virus. So you may be thinking to yourself, hey, are interferons being researched right now as a way to treat viral-based infections? And the answer to that is absolutely. Right now, antiviral research into interferons is red hot in the biomedical field. Here's a diagram for your text illustrating the complement protein process. We are first going to have some complement proteins bind, and they poke holes in the bacterial cell membrane. These holes are called an MAC, a membrane attack complex. Oh, I love that name. That is a very cool name. And once we have lots of these membrane attack complexes formed within our bacterial cell membrane, we are going to have water rush in, and then the bacterial cell will pop like a balloon. So, let's have a concept check question. Which of the following does not apply to innate defenses within our body? We have protects against a broad range of microbes, inflammation in the complement system, physical barriers, or targeting a specific pathogen. And when I say a specific pathogen, I mean a specific species of bacteria or virus. So go ahead and pause this video and get me an answer. 5, 4, 3, 2, 1. All right, the correct answer is D, targeting a specific pathogen. Our innate or non-specific immune system is going to protect against a broad range of microbes. It includes the inflammation response and the complement system and many physical and chemical barriers. Our innate or non-specific immune system does not target a specific pathogen. If you have any questions for me about the non-specific or innate immune system, please feel free to post them on the class discussion board, shoot me an email, or swing by my office when you're on campus. Happy studies!